On Friday, we left off here kind of talking about tubular reabsorption. Okay, so this is one of our 3 steps related to urine formation. We discussed the first step, which was glomerular filtration, where we pulled out the water, the ions, some waste, some glucose from the blood, and we filtered that into the renal tubule. Okay, now we're going to put a lot of that stuff right back in through this process. Okay, tubular reabsorption is where we return those things back to the blood, and we showed that here with these figures, which can be done 2 different ways: paracellular, transcellular. Paracellular, remember, refers to traveling between the cells, whereas trans crosses through the cells, from the apical membrane out to the other side, to what's called the basolateral membrane, on its way through the bloodstream. Okay. Now, sodium is shown here, and remember, we talked sodium as being the most important of the substances that's going to be moved out of the lumen. Okay. So we did discuss this stuff here, right? Okay, so just refreshing on that. As sodium is pulled from the lumen of the tubule to the blood, glucose can hitch a piggyback ride and be brought into the tube cells. Okay, so that's an example of cotransport. Okay, hydrogen ion does the opposite. It's going to run through an antiporter. Okay, and so it's going to go into the lumen. Okay, and then when we talk water and various solutes like chloride, potassium, calcium, magnesium, and urea, these things will move either through the cell, through transcellular or paracellular, between the cells, and then into the blood, and water will chase after it. Okay, so water will always be coupled to the movement of various solutes. Okay. Now, water is reabsorbed in high quantities right away. Okay. We said 99% of the stuff or 99% of the water that was brought into the tube was pulled right back into the blood. Okay. A lot of that percentage comes directly in that first segment of our tube, which was the proximal convoluted tubule. Okay. So now we'll move from the proximal convoluted tubule to the loop of Henle, which, if you guys remember, is the segment of our nephron that dips down into the medulla. A pretty complex but cool process takes place here. It's called the countercurrent multiplier mechanism, and I'll show that here in just a second. As I said, most of that water that was pulled out of the blood gets put right back into the blood in our proximal convoluted tubule, about 65% of that water. So the fluid that enters the loop of Henle is very, very different chemically than the water or the fluid that was entering now into the proximal convoluted tubule from the glomerulus. Okay. Loop of Henle continues the reabsorption of water, about 50%, and solutes like chloride, potassium, magnesium, calcium, and bicarbonate. For the first time, however, water is not coupled to the movement of those solutes because the part of the loop is relatively impermeable to water. And the part that is relatively impermeable to water is the thick segments. Okay, and that's what we're going to show here on this page. Okay, so here we can see where we first formed the filtrate back in our renal corpuscle, and that filtrate's then traveling through our proximal convoluted tubule. In our proximal convoluted tubule, we said that water, about 65% of that is actually brought right back into the bloodstream. Okay. So the water that's found along with the other substance inside the tubule here in our loop of Henle is a lot different than the fluid that was back up here. Okay. We're going to continue to reabsorb water, and that's why you can see here an arrow of water going out of the tubule. Okay? Why this water is reabsorbed here is because of what's happening over here in the other side of the loop of Henle. We have the descending limb on the left here and the ascending limb over here on the right. Okay, if I go back, tubular reabsorption and secretion in the loop of Henle occurs by way of a countercurrent multiplier mechanism. The first of the interactions between the descending and the ascending loops of Henle. It operates to increase concentration of the renal medulla, and we reabsorb water from the tubule by way of osmosis in the descending limb, what I just showed you in the figure. This is because the interstitial fluid of the renal medulla is very hypertonic. Okay. The fluid that is found here in the renal medulla is extremely hypertonic. Okay. The larger the number, the higher the osmotic gradient, which means the more water basically wants to travel there. If you guys remember when we talked about hypo, isotonic solutions all the way at the beginning of the semester, we learned that salt water, for example, is very hypertonic, which meant that there was a lot of solutes in it. And if you drink that, the water that's in your cells is going to travel to where that salt is. So wherever there's a lot of solutes, that's going to be hypertonic, and therefore water is going to flow in that direction. Okay, that's what happens. And the reason that this interstitial fluid is hypertonic is because of what we're doing in this ascending limb. In the thick segment of the ascending limb, we're pumping out sodium chloride. What is sodium chloride again? Essentially the form of, or known as the form of, salt. They say if you were to taste the middle of a kidney, it would actually taste really salty. Okay. Now the water goes to where the salt is. So we pump out the salt over here, which causes the pulling out of the water over here. Both of those processes are showing reabsorption. Okay. So that's why this is showing, as the title of the slide, reabsorption. Okay. Now, the water that's pulled out of the renal tubule in the descending limb will not stay in the renal medulla. There is a set network of blood vessels called the vasa recta that surrounds that loop of Henle. It allows the water to enter the blood directly and be carried away. So the water never helps to achieve osmotic balance. Okay. We're doing to another thing here is urea, and we're going to perform what's called urea recycling. Okay. And if you pay attention to the figure, it'll show that pretty well. Okay. Right down here at the bottom of our figure, we're showing off urea. Okay. Urea gets recycled because we secrete it, which means it goes into the tubule in the loop of Henle. That urea will stay in the tubule past the DCT, then down the collecting duct, where we then reabsorb it. Notice how the DCT, or the collecting duct, runs right next to the loop of Henle. So urea gets pulled out, and then put back in. And then it gets pulled out, and then put back in. So now hopefully you can see why we call it recycling. Okay. Does that make sense? So we have this crazy process that's happening between our descending limb and our ascending limb. Pumping out salt, pulling out water. Okay? Pumping out salt in the ascending limb, pulling out water in the descending limb. Okay? And then urea is recycled between the loop of Henle and the collecting duct. Okay, last segment of the nephron is the distal convoluted tubule, and then we can also throw in the collecting duct. So as you can see up there at the top, we have DCT and then CD. Those are short for distal convoluted tubule and collecting duct. Here, we're going to be sensitive to hormonal influence. Okay, you can see there are several hormones. We've already talked about some of these in this lecture. We've definitely talked about all these hormones at various points in the semester. All right, if I go back to this figure, I can kind of illustrate what's going to happen. Okay, so back here in our loop of Pinlay, we suck out, and I shouldn't really say suck out, I pumped out, or we pumped out salt from our ascending limb, which means the fluid inside of our tubule is now pretty hypotonic, which means it's low in solute number. Basically really watery, okay? And in that case, what happens is water is going to leave it, which we can illustrate here with our nice arrows right there by H2O, okay? That's going to help us to perform what's called water conservation, okay, to save water from being peed out, okay? So since salt is pumped out of the filtrate, the ascending loop, fluid in the DCT comes hypertonic. Interstitial space of the renal medulla, as we mentioned, is very hypertonic. This helps form the osmotic drive for the reabsorption of water across the walls of collecting ducts, allowing for some water conservation. So this doesn't just happen up here. It also happens, as we can show down here, with our collecting duct. Okay, H2O is leaving to enter into the interstitial space. Okay. We have what are called principal cells that are found in the distal convolute tubule and the collecting duct, carrying receptors for specific hormones, regulating amounts of water and salts reabsorbed. The first hormone that we are going to talk about is angiotensin II. We learned the other day that it reduces the formation of filtrate, so it reduces GFR. It also enhances the reabsorption of sodium chloride and water in the proximal convolute tubule and stimulates the release of aldosterone. Okay. Aldosterone is our salt-retaining hormone, so if we release it, we're going to retain salt, sodium chloride, and water, therefore reducing urine volume. And as we learned the other day, if there's more blood volume, there's more blood pressure. Okay, this is all straight up stuff we talked about the other day. HO natriuretic peptide, remember, was the one that was produced by the cells of our heart in response to too much blood. And this is going to cause the excretion of more salt and water in urine, which reduces blood volume, therefore reducing blood pressure. This is the only one that's going to make us pee more water and salt out. Okay. The others are going to help to conserve it. Okay. So this is the only one that increases excretion, whereas the other ones are going to help conserve it by reducing excretion. Antidiuretic hormone is our water conservation hormone secreted by our posterior pituitary when our body becomes dehydrated and our osmolarity in our blood goes high. Stimulated to be released by osmoreceptor neurons in the hypothalamus. When it increases in plasma, osmolarity increases like in dehydration. Overall, what you'll want to know is that it promotes water reabsorption. Okay. So promotes reabsorption, reabsorption, reabsorption. This is the only one that promotes secretion. Okay. The last one has nothing to do with water or salt. It is related to blood calcium. We release it when our calcium levels are low, what we call hypocalcemia, and therefore it promotes the reabsorption of calcium, which means we're going to lose less calcium out in our urine. Okay. Make sense? You want to know what the overall effect of these various hormones is. Okay. There's a nice table here on the next page that even gives it to you. So, there you go. Any questions on anything so far? Here's a nice summary of all the things that are going to be done in the formation of urine. Okay, in our corpuscle, we form the filtrate. We talked about GFR. Okay, what's brought out? Okay, not plasma proteins, blood cells, or anything like that. Okay, then we perform reabsorption. Primarily, water we see is brought back into the blood from the proximal convoluted tubule. We didn't talk about it, but we do secrete in the PCT. Okay, not super important stuff though. I mean, this is important to get rid of, but we didn't really talk a lot about it. We did talk about hydrogen ion though, that it is brought into the tubule. That was not the only thing. In our nephron loop or loop of Finlay, we talked about that countercurrent multiplier mechanism, and then the last thing we talked about was reabsorption in the DCT and the collecting duct. And we didn't really get too much into secretion there, but that does happen as well. You won't need to know this figure or anything crazy like that, but you know it's kind of a helpful thing because it shows what's happening and where. So. All right, now we've made the urine. We then must move it from our kidneys and get it ready to be moved out of the body. Within our kidneys, we have the renal pelvis, which is an extension of the ureters, which are muscular tubes passing dorsal to our bladder and actually draining into it from below. Our bladder is a muscular sac on the floor of the pelvic cavity, inferior to the peritoneum, posterior to our pubic symphysis. It is made up of 3 layers. The middle layer is a muscular layer made up of a muscle called the detrusor muscle, very important for urination. Okay. Openings of the 2 ureters along with the urethra form a triangular-shaped trizone, which is a common site of bladder infection. This is more anatomy-based stuff. But what is more physiology is the 2 sphincters that guard the urethra, which is the tube that carries the urine from the bladder to our external urethral orifice. We have an internal and an external, very similar to how we had an internal and external anal sphincter. What's even nicer is that it's the same thing. The internal is involuntary, external is voluntary. And it has everything to do with the type of muscle. The internal is made up of smooth muscle, whereas the external is made up of skeletal muscle. The same thing was seen in our anal sphincter situation. Internal was involuntary there, external was voluntary there. Same thing exists. Okay. Now, anatomically, the urethra is very different between men and women for a variety of reasons. There's 3 parts to a male urethra. It's much longer. It's a shared tube, so we'll actually talk more about it in our next lecture. In females, it's real short, and it's a singular tube only carrying urine. That's its only function. And in men, it carries not only urine, but also semen. And we'll talk about that in our next lecture. The actual act of urination is called micturition. Partially controlled by a spinal micturition reflex in a reflex center of our sacral region of the spinal cord. So this involves the last little bits of the spinal cord and involves a process that can be illustrated here on this next page. So the reflex is initiated when the bladder begins to fill up with urine. There are stretch receptors that are found within the wall of the bladder, kind of like how we saw stretch receptors were found in the stomach, for example. And as the urine fills up the bladder, the stretch increases, which activates those stretch receptors that send signals into the spinal cord. Okay? And that's what we're showing here with step 2. Step 1 was just filling up the bladder with the urine. Okay? The third step is parasympathetic nerve activity causing contraction of the detrusor muscle, and we see relaxation of the internal urethral sphincter. Okay. Because at this point, Well, hang on, I'll go back. Let me go back before I do that. Okay. We contract the detrusor. We relax the internal urethral sphincter. At this point, we have the urge to urinate. But our external urethral sphincter will help prevent that from happening until it's, a proper time. Okay. We will still have conscious control over that. And when we want to actually urinate, we will send descending motor tracts to the micturition center that produces inhibition of the somatic motor neurons in the external urethral sphincter, which causes it to relax and therefore urination to occur. So what happens is we have signals that come down from the brain and signals that come from the spinal cord that cause the external urethral sphincter to relax. So we inhibit that muscle, it relaxes, and we urinate, or we micturate. It's important that you do not confuse the word micturition with mastication. They sound similar. What was mastication? Anybody remember? Chewing. So very, very different than peeing. All right, last thing I want to talk about in this lecture is acid-base balance. Something that we've talked a little bit about the last couple of weeks. We've even talked all the way back at the beginning of the semester about the importance of a proper pH. Okay, major homeostat challenge is keeping our hydrogen ion concentration or our pH of our body fluids at an appropriate level. Without the maintenance of acid-base balance, normal cellular function would not exist. Remember, our proteins prefer a specific pH and temperature, keeping them from denaturing, where they unwind and lose function. We're going to focus on the pH of blood, because the pH of blood really kind of affects the pH of every other fluid. We want our pH of blood to be 7.4. Okay, that's its perfect value, its set point. Anything above 7.45 is going to be bad. Anything below 7.35 is bad. So this window between 7.35 and 7.45, with 7.4 being right smack dab in the middle, is our narrow limits of survival. Does our body ever get outside of that window? Yes. Okay. We'll get to that in just a second. In order to keep our body at that hopefully 7.4, we depend on three major mechanisms for maintaining that pH. The first is a set of buffers called buffer system, acting quickly to temporarily bind to hydrogen ion and remove it from solutions. This will help raise pH of body fluids, but won't remove hydrogen ion from the body. The most abundant buffer in our intracellular fluid and blood plasma is the protein buffer system. Proteins are made up of amino acids. So the amino acids making up those proteins are capable of releasing hydrogen ion when pH rises, and this can then combine with hydroxide forming water. The other end of the amino acid is capable of combining with hydrogen ion when pH will fall. The carbonic acid bicarb acid buffer system we've talked about before is based on the bicarbonate ion and carbonic acid, which obviously is an acid. Okay, we can control those levels based on the pH of the body. And then the phosphate buffer system utilizes various phosphate ions, regulating acid-base balance in our cytosol. In our last unit, we talked about breathing carbon dioxide. If we increase the breathing of CO2, it's going to help bring our pH which direction? It's going to cause it to go up. Okay, that's what hyperventilation is. We know if we have CO2 in our body, it will combine with water to form an acid. So by getting rid of more CO2, there's less acid, pH goes up. If we hold our breath like in hypoventilation, or our breathing rate slows down, more CO2 will be retained in the body, and that will cause your pH to go down. This is important in short-term pH mechanism, or pH balance, because we breathe second to second. Okay. Something that's brand new to us in this lecture is how our kidneys excrete hydrogen ion to get it actually out of the body. This is the slowest mechanism, but it's the only way to eliminate acids other than the carbonic acid that we were just talking about. And this acid is going to be lost through urine. So if you were to take a pH sample or a sample of urine and perform a pH test on it, you would find out that it would be, or should be at least, slightly acidic. Now I said sometimes our body will illustrate situations where we're outside of that 7.35 to 7.45 window. When our pH drops below 7.35, we call it an acidosis. Remember, 7.35 is still technically basic because 7.0 is neutral and anything above that would be a base. But we call it an acidosis because of the buildup of acids in the body. Major effect of this would be depression of the central nervous system through depression of the synaptic transmission. So we have less synapse formations, okay, or synapse signals. This can cause a person to fall into a coma, and that could lead to possible death, or could cause possible death. Alkalosis is the opposite. Blood pH goes above 7.45. This causes things like overexcitability in both the central nervous system and the peripheral nerves, causing nerve impulses to be conducted repetitively even when not stimulated, causing things like nervousness, muscle spasms, and even convulsions and death. Okay. So, not good to have either of these conditions. In order to counter alkalosis or acidosis, we perform what's called compensation. And there's two forms. Respiratory compensation, where we change our breathing rate. Whereas that's more important in quick restoration, we also have renal compensation, which is more of the long term, as we've talked about. Okay. Respiratory compensation involves changing the respiration to regulate how much PCO2 we have in our blood. Okay. Any increase in blood PCO2 will lower pH, whereas a decrease in it will increase pH. We've talked about that before. Okay, that's pretty easy to illustrate. Okay, and that's what we're looking at over here with this feedback loop. Okay, we have a decreased pH, okay, because hydrogen ion is increased. The receptors we learned are peripheral and central. We've talked about this before. Output causes us to breathe faster. More CO2 is exhaled. pH will go up, and therefore we return back to normal. Negative feedback in a nutshell. Okay. Renal compensation is utilizing the kidneys through the blood bicarbonate concentration, reducing the rate of bicarb excretion when pH decreases, or by increasing the rate of bicarbonate excretion when pH increases. Remember, bicarb is a base. And if you combine a base with an acid, they neutralize each other. So if our pH drops, we're becoming acidic. In order to counteract that, we want to maintain or keep our bicarb levels high. And if our pH goes up, we want to get rid of any bases to make it go down. And hopefully you guys can follow along with this stuff here on this last page, kind of illustrate what happens. It's a pretty cool process. We have 4 conditions of acidosis or alkalosis that exist. Okay, and then we're going to talk about the compensatory mechanism. Down here at the bottom, we've got a figure to illustrate, and this isn't important, I promise you, for the test, but I want to illustrate what happens. We've got what occurs when we see an alkalosis or acidosis happening. Okay, so point A in the middle of our figure is where our body wants to be. We've got 3 different conditions that we're plotting on this graph. PCO2, we want our blood PCO2 to be at 40. Our pH, we want to be at 7.4, and our bicarb, that's what HCO3 is about 26. Okay, this is where we want to be. Those 3 things right there are in perfect balance. Okay. During a respiratory acidosis, we increase PCO2, which drops our pH. Okay, which, if you pay attention, respiratory acidosis is like going from A to B. Because our PCO2 levels go up, our pH goes down. How do we accommodate that? We compensate with our kidneys by conserving bicarbonate. So we go from that 26 all the way up to 40. So this is A to B to D. Our PCO2 is not changing, our bicarb is, but the most important thing is we're ending back up at 7.4. Okay. The opposite occurs with respiratory alkalosis. PCO2 drops from 40 to 20, our pH goes up. So our body will compensate by getting rid of bicarbonate. And that level is going to drop. We end up back at 7.4. And metabolic acidosis is like going from A to B, or A to G to F. Our PCO2 is not changing, it's our bicarbonate. Okay? This can happen like during diarrhea. Okay? So our bicarb drops, our pH drops, we compensate by breathing faster or hyperventilating, and we return to 7.4. Okay. Metabolic alkalosis is shown right here. Okay. PCO2 doesn't change, but bicarb levels go way up. This can happen like vomiting can cause this. This time we're going to hypoventilate to go to 60. pH goes back to 7.4. So notice that the goal overall is to always get back to 7.4. It's just how we get there that's different. And any questions on that? Like I said, we don't need to really worry about this or this. This figure is just showing out how we can get hydrogen into the lumen of the tubule to get it out of the body. And this over here on the right is just showing how pH can drop or increase. Pretty straightforward. Either loss of bases, increase acids, or vice versa. All right, any questions on anything on the kidneys?